right, hello everyone. Thank you very much for coming to our August session of the Medical Mycology Trainee Seminar Series. To start with announcements of interest to the community, the monthly Mycotalks is this month on the 26th of August with two really great speakers, Bridget Barker and Frank Vanderdonk. And so I'll put the information on registering for that into the chat, but I highly recommend that you all check that out. And our first speaker today is Shelby Priest. And Shelby attended Rice University in Houston for her Bachelor of Science degree, where she started on fungi by investigating the CUG clade under the mentorship of Mike Lorentz. She then moved to Duke as a PhD student, where she joined Joe Heitman's lab and has been studying pathogenic Cryptococcus species. She's been the recipient of an NIH F31 award, and her paper was just posted on bioRxiv. So we're in for a fabulous talk from Shelby today. Shelby?
All right, thank you, Jessica. I'll go ahead and share my screen and get it going. Okay, so hi everyone. Like Jessica said, my name is Shelby Priest, and I'm a grad student in Joe Heitman's lab. And today I'm going to be telling you all about an interesting mechanism of drug resistance that we discovered in Cryptococcus Neoformans and tell you about some of the genetic insights that we've gained behind this mechanism of drug resistance. So Cryptococcus neoformans is a human fungal pathogen that is ubiquitous in the environment. It's commonly found in pigeon guano and soil, and spores and desiccated yeast cells from the environment are regularly inhaled into our lungs. Like most deadly fungal pathogens, Cryptococcus typically only causes infections in immunocompromised individuals, causing pulmonary infections and eventually migrating to the central nervous system to cause cryptococcal meningitis. Each year, there are over 200,000 new infections, and the standard treatment strategy for cryptococcal meningitis is amphotericin B therapy, ideally in combination with 5-FC, followed by fluconazole maintenance therapy. Unfortunately, 5-FC monotherapy cannot be used alone due to the development of high levels of resistance, and Cryptococcus can also acquire resistance to fluconazole. Patients with cryptococcosis face unacceptably high mortality rates, ranging from 20% in those receiving treatment in more economically developed countries to 100% mortality for patients not receiving treatment. As a whole, cryptococcal infections account for approximately 15% of AIDS-related deaths. So this project started off with the initial question, do hypermutators of Cryptococcus neoformans exist in nature, given the high levels of resistance we see to current antifungal therapies? Before we begin, I wanted to clarify that for our study, we're defining a hypermutator as a strain with an increased propensity to develop de novo resistance to antifungal drugs, compared to both closely related strains, as well as the laboratory reference strain H99. Fortunately, right when this project started, whole genome sequencing for a collection of nearly 400 Neoformans clinical and environmental isolates had become publicly available. And all of those strains in the collection were also housed at Duke. So all of those isolates are what is pictured in the phylogenetic tree you see here on the right. So we began looking for hypermutator strains with a basic assay where we grew independent overnight cultures of each strain, swabbed them to antifungal drug media, either YPD supplemented with rapamycin and FK506, or YNB supplemented with 5-FC, and strains that produced more colonies on average than the reference strain H99 were categorized as hypermutator candidates. Through this method, we were able to identify 40 hypermutator candidates. One interesting side note is that out of the 14 environmental isolates we screened, 13 of them came up as hypermutator candidates. But following this screen, we chose to focus on 2 of our strongest candidates on YPD supplemented with rapamycin FK506, which were BT65 and BT81, which you can see pictured here on the plate, along with our negative control H99 and our positive control, which is a deletion mutant lacking the DNA mismatch repair gene MSH2. And BT65 and BT81 are 2 very closely related clinical isolates. After identifying BT65 and BT81 as hypermutator candidates, we wanted to confirm that they were indeed hypermutators by accurately quantifying their mutation rates through fluctuation assays, which allow us to measure the number of cells in the culture and also measure the number of spontaneously resistant cells that arise in the culture. Here we looked at the mutation rate on rapamycin FK506. First, we have our negative control H99, our positive control, again, the MSH2 mutant. And then we have, in addition to BT65 and BT81, 11 of the most closely phylogenetically related strains. And as you can see here, we confirm that both BT65 and BT81 are indeed hypermutators. So after this confirmation, we wanted to determine the genetic basis of this hypermutation. Fortunately, when using the combination of rapamycin and FK506, mutations in only a single gene can confer resistance, and that gene is FRR1. So we started off by looking at resistant colonies from our control, H99, and saw a lot of the expected PCR product sizes, which are about 1.2 kb, with the exception of a few smaller products, which are deletions. However, the FRR1 PCR products from resistant colonies of BT65 and BT81 were surprisingly larger than the expected product size a lot of the time. We quantified these changes and sequenced these PCR products and found that a non-LTR retrotransposon known as C. Neoformans line 1 or CNL1 was responsible for conferring the majority of resistance in BT65 and BT81. And we also saw that every single insertion in FRR1 in these two strains was caused by CNL1. This was particularly surprising because Cryptococcus neoformans was not previously thought to encode any functional copies of CNL1. And typically all other transposons in Cryptococcus are silenced by RNA interference. These results in BT65 and BT81 were obviously in stark contrast to the mutation spectrum we saw in H99, which were all SNPs, deletions, or microindels. And two important things I want to note are that one, we didn't observe CNL1 insertions in drug target genes in any of the other closely related strains. And two, that CNL1 insertion could also confer resistance to 5-FOA via insertions in the URA5 gene, as well as to the clinically relevant antifungal drug 5-FC via insertions into the FUR1 gene. However, BT65 and BT81 weren't hypermutators on those media. So after determining that CNL1 was responsible for resistance to all of these different antifungals, we wanted to know what was allowing these elements to move in the genome since they're normally silenced by RNAi. To accomplish this, we generated 28 progeny from a cross between BT65 and H99. And here you can see we found that they displayed a wide range of mutation rates. And you can see all of their mutation rates here compared to the H99 parent and the BT65 parent. So we subsequently sent these progeny for whole genome sequencing so that we could conduct quantitative trait locus mapping or QTL mapping. So QTL mapping works by identifying all of the genetic differences between the two parental genomes and then looking to see if inheritance in the progeny of any of those differences at specific loci are more likely to be associated with a hypermutation phenotype in the progeny. This lovely GIF was made by our collaborator Colin Roth and shows you that QTL mapping in the 28 progeny that we sequenced identified two significant QTL peaks on chromosomes 3 and 11. And in the top part here, you can see just how strictly these two loci are associated with the hypermutator and non-hypermutator phenotypes based on which parent they are inherited from. So here again, we have the mutation rates of the progeny along with a stable figure of the QTL mapping. And you can see again, those two significant QTL loci on chromosomes 3 and 11. And what was really interesting that happened next was that we identified a nonsense mutation in the first exon of a gene encoding the RNAi component CNF3, which is known to silence transposons. And that was located under that chromosome 3 QTL peak that was inherited from the BT65 parent. And when we looked back at the progeny and looking at which ones inherited which ZNF3 allele and which ones are hypermutators, you can clearly see that the ZNF3 nonsense mutation from the BT65 is associated with higher mutation rates. So now you might be saying to yourself, wait a second, I see two QTL loci here, this one under chromosome 11. there must be another locus contributing to hypermutation under that second peak. But it turns out that these two QTL peaks actually represent a single genetic locus because there's a chromosomal translocation that differs between the two parents, which I have depicted here in this chromosomal schematic we made based off of nanopore long-read whole genome sequencing. And moreover, if you look at the genetic loci inherited in the progeny from either parent, here we have loci inherited from the H99 parent in blue and loci inherited from the BT65 parent in orange. And then along the y-axis, we have the progeny sorted according to their mutation rates with all of the hypermutator progeny above this dashed red line. And you can see that the two QTL loci in orange, here and here, are always inherited together, and that they also include that chromosomal translocation. And you can also see just how closely associated that ZNF3 allele is with that translocation. So identification of that nonsense mutation in ZNF3 was very exciting because our lab had actually previously discovered the role of ZNF3 in RNAi and in transposon silencing. And moreover, this SNP causing the nonsense mutation was exclusive to both BT65 and BT81 and not found in any other closely related strains. And this mutation is predicted to lead to a severely truncated version of this protein, suggesting RNAi might not be functional in BT65 and BT81. So because of the identification of this mutation and the fact that CNL1 transposon was mediating the majority of resistance, we conducted nanopore long-read whole genome sequencing that I had mentioned earlier to further characterize the distribution of transposons in the genomes of these two hypermutators. So nanopore sequencing revealed that both BT65 and BT81 encode several hundred copies of CNL1, indicated by the black and gray boxes in the figure here. And you can see that the CNL1 elements primarily localize into arrays at subtelomeric loci. This localization is likely due to the fact that CNL1 is hypothesized to preferentially insert at the telomeric repeat sequences of Cryptococcus neoformans. And these results were especially shocking because, like I also mentioned earlier, the laboratory reference strain H99 doesn't encode any full-length copies of this transposon and only encodes 22 fragments of CNL1. And you can see here that same figure from the last slide, and now we're including what the genome of H99 looks like. And you can see just how stark the contrast is between H99 and the two hypermutators with respect to their CNL1 burden, as well as the localization of these elements into the subtelomeric loci. Interestingly, we didn't notice any significant changes when looking at transposon burden of any other transposons in the strains compared to H99. So one interesting phenomenon in Cryptococcus is that we know transposons are highly upregulated during mating. So to look at the dynamics of CNL1 during mating, we conducted nanopore long-read whole genome sequencing of a subset of those F1 progeny that we used for the Qtl mapping. And at the top, we have pictured the whole genomes of the parents with H99 all in blue, BT65 all in orange. And then you can see the recombination maps of the progeny, showing that they are indeed genetically recombinant progeny. And so below, if we look more closely at some of the subtelomeric loci in the progeny, we also found what appears to be rapid expansion and contraction of the subtelomeric loci, as well as invasion of CNL1 into subtelomeric regions from H99 that previously had no copies of CNL1. And we also call these subtelomeric regions from H99 that had no copies naive subtelomeres. And you can see in the subtelomeres pictured here that every single one, we have an example where the CNL1 elements from BT65 have invaded one of these blue regions from H99. So finally, we wanted to determine if only that single SNP causing the ZNF3 nonsense mutation was responsible for hypermutation in BT65. We identified an allele of ZNF3 in the closely related strain BT133 that had no mutations within 1 kb on either side of the nonsense mutation compared to VT65, other than that nonsense mutation itself. And we use CRISPR-mediated transformation to create a double-stranded break at that nonsense mutation, and then provided the BT133 ZNF3 allele for homologous recombination. We identified successful transformants, assayed their mutation rates, and found that replacement of the ZNF3 nonsense mutation in BT65 significantly reduced the mutation rates in 2 independent transformants. We also characterized RNAi function in BT65, BT81, and the complemented strains by analyzing their small RNAs. We found that a ZNF3 deletion mutant in H99, which is pictured here, as well as BT65 and BT81, produce small RNA size distribution profiles identical to that of an RDP1 deletion mutant, which is pictured here in the dark orange, which lacks an RNA-dependent RNA polymerase required for RNAi. And we also showed that ZNF3 complementation restored production of the canonical 21 to 24 nucleotide small interfering RNAs involved in RNA interference. Ultimately, we also showed that ZNF3 complementation restored production of small RNAs corresponding specifically to CNL1 elements in BT65. And here we have pictured here the sense and antisense small RNAs corresponding to an array, one of those subtelomeric arrays of CNL1 elements on a chromosome in BT65. And you can hopefully see BT65, which is the flat green line pictured here at the bottom for both the sense and antisense small RNAs, showing that BT65 essentially produces no small RNAs corresponding to CNL1 elements, which is very different from what we observe in the 2 ZNF3 complementation mutants, which are in the dark blue and light blue lines pictured here. So with that, I'd like to conclude my talk and say that I've shown you that BT65 and BT81 are both hypermutator strains of Cryptococcus neoformans, that the non-LTR retrotransposon CNL1 mediates the majority of FK506 and rapamycin resistance, but also that CNL1 can mediate resistance to 5-FOA and the clinically relevant antifungal drug 5-FC. I've shown you that BT65 and BT81 encode hundreds of subtelomerically localized copies of CNL1, and that these elements are also capable of invading naive subtelomeric regions in a non-hypermutator strain. That RNAi loss in BT65 and likely also BT81 is due to a single nonsense mutation that's caused by a single SNP in the RNAi gene ZNF3, and that replacement of the ZNF3 nonsense mutation in BT65 with the wild type SNP significantly reduces mutation rates and restores RNAi function, including small RNAs corresponding to CNL1. Future directions for this project include determining if CNL1 insertion can confer resistance to the other 2 antifungal drugs used in treating Cryptococcus, which are fluconazole and amphotericin B. We'd also like to look at the effects of hypermutation in vivo, looking both with and without antifungal drug treatment to see if CNL1 could confer resistance in vivo. And then finally, we'd like to look at the impacts of the CNL1 movement and hypermutation on overall fitness and genomic stability over time. And so with that, I would like to first and foremost thank my mentor, Joe Heitman. I'd also like to thank the Heitman lab members, specifically my collaborators for this project, which have been Vika Asiatov, a postdoc in our lab, as well as Colin Roth and Tim Dahlman. And you can also follow our lab on Twitter if you'd like. And I'd also like to remind everyone that the manuscript for this project actually just went live like at midnight last night. So it's very exciting. And if you want to learn more about it, it's on bioRxiv now. And so thank you guys all for listening. That's it.
All right, thank you very much, Shelby. That was fantastic. We have a few questions in the chat already, but please enter any questions you have in the chat, and then we'll take them after both speakers have presented. And so now I'll turn it over to Liz and Herbert, who will introduce Bing.
Hi, everyone. I'm stepping in for Herbert. He's having internet issues. But just to say thank you so much to Shelby for a fascinating, interesting talk. I'm sure we'll have a lot of discussion after Bing's talk. It's my great pleasure to introduce Bing, who received her BS in Biological Sciences from China Agricultural University. She then earned her PhD in Zhai Lin's lab at Texas A&M. And for her postdoctoral studies, she joined Tobias Hull's lab at Memorial Sloan Kettering Cancer Center in New York City. So she's been all around the world. She's recently started her as a principal investigator at the Institute of Synthetic Biology at the Shenzhen Institute of Advanced Technology and the Chinese Academy of Sciences. And I think we're really in for a treat to hear about what her lab is going to be doing. I know she's going to be advertising a position shortly, and I think she'll talk about that a little bit at the end of the talk, but this is your chance if you want to know what that opportunity might look like. Take it away, Bing.
Okay, thank you very much for the introduction, Liz, and thanks for the invitation, Jessica. It's my great honor to give the presentation here. It's very weird to say good morning in the middle of the night in China here. But yeah, as Liz just said, I recently came back to China to start my own lab in the Shenzhen Institute of Synthetic Biology. As of today, my lab is 5 weeks old. So I'm going to present the data that I generated in Memorial Sloan Kettering in Tobias lab. I graduated from China as an undergraduate student and came to Texas. And I had my PhD in Dr. Sharon Lin's lab and studied the biology and pathogenesis of the human fungal pathogen Cryptococcus Neoformans. And when I'm looking for postdoc positions, I contacted Dr. Tobias Hull in Memorial Sloan Kettering Cancer Center. And when I contacted Tobias, I thought he's going to assign me to a Aspergillus immunology project because that's what he established his lab. But when I interviewed with Tobias, he said he wanted to study new directions in the labs to study the intestinal microbiome. I thought this is a very interesting research directions, and I took the risk and became the outlier in the lab in the next 4.5 years. But now you can see the result is pretty good. And I really appreciate my two mentors, Sharon and Tobias, for their guidance in my research in medical mycology field. And I think I'm way beyond lucky to have both of the two fantastic mentors in my studies. And as I know that both of the labs are hiring, so if you graduate students, you're very interested in fungal biology or fungal immunology, please contact them. Okay, let's back to my topic today, the human microbiome. In the year that I joined Memorial Sloan Kettering, there is a very important review article from Drs. Elaine Elias and David Enderhills that discussed about the human microbiomes on different sides of the mucosal surface. Apparently, we can find fungal floras in all this kind of different sides of the mucosal surface. But there are not many labs, even till now, there are not many labs in the world to study the microbiome, the fungal flora. And a very common question I was asked when I gave talks outside the mycology world, they will ask, are there fungi in the gut? So why people don't appreciate the significance of fungi in the gut? That is probably because that in healthy people, there's not a lot of fungi in the healthy people's intestines. So this is a study from Jeff Gordon's lab that they did a very nice survey study of from the fecal samples from different people with different geographic origins and different ages. And you can see that no matter where are the people from and no matter how old are they, the abundance of fungal DNA in all the fecal DNA samples remains very low. So it's like 0.01 to 0.1%, which means the abundance of fungi is about it's about 3 or 4 logs lower than the bacteria. And a very interesting study recently that this is a study from Joe Petrosino's lab in Baylor. What they have is here. So we know that Candida albicans is an oral cavity commensal. And actually, every time when we swallow saliva or when we eat, we kind of inoculate ourselves with Candida albicans. What they did is this. So they asked a volunteer to use different teeth brushing protocols. It's either once a day or with increased teeth brushing that every time after the eat, they ask the volunteer to brush their teeth. So these different tooth brushing protocols can change the abundance of oral cavity colonized Candida albicans. Therefore, what they read out is the fecal Candida burdens. And you can see that as the change of the teeth brushing protocols, there is a fluctuation with fluctuation of the fecal fungal Candida burdens. This suggests, therefore, they speculate that the gut fungi is rather a passerby instead of stable colonizer. But with all these controversies, when I started my research in Device Lab, there are 3 keywords and related questions about the gut microbiome: the context, approach, and the implication. So in which conditions can I observe a stable microbiome signals? And how can I get reliable data? And how can I interpret the data? How can I use the microbiome data to help improve the approaches and help us to understand the different pathological disease contexts? So my talk will basically follow the 3 keywords. And I want to introduce 2 fecal samples to the audience. So these 2 fecal samples were collected from the same individual. Sample B was collected 12 days after sample A. And through the fungal ITS sequencing, you can see there's this big change of the fungal composition. That is, the sample B has a significant expansion of the Candida parapsilosis. Meanwhile, the sample in the bacterial compartment of the same 2 samples, we observed a reduction of the lactose from the sample A to sample B. The lactose is one of the core taxa of the anaerobic bacteria and is considered as a hallmarker of the healthy microbial flora. So these relative abundance of the microbial components analysis suggests that there are composition changes of the fungal compartment and bacterial compartment respectively. But what about the fungi compared to the bacteria in the same sample? Luckily, we have the shotgun data of the 2 samples. And what we found is that in sample A, the non-fungal component, which are mainly bacteria, is about 4 to 5 logs higher than the fungal reads. And in the sample B, where has Candida parapsilosis expansion, the reads between bacteria and fungi are almost similar. Therefore, we can see that the expansion of the Candida species coincides with the loss of the bacterial abundance. What's more interesting is that 2 days after the sample B being collected, this patient was found to have the Candida parapsilosis bloodstream infections. From this example and a lot of other studies, people now start to speculate that the intestinal fungal expansion is maybe a kind of a sign of the dysbiosis of intestinal microbial floras. And how this dysbiosis happened, in terms of this patient, this is a allogeneic hematopoietic cell transplantation patient. So this allo-HCD procedure, although it's a curative therapy for a lot of hematopoietic malignancies such as leukemia, it also creates highly immunocompromised conditions and can damage the barrier integrity of the mucosal surface. Therefore, these patients are at high risk to develop a lot of different infections, and they are required to have prophylactic drugs, which in turn changes the dynamics of the intestinal microbial flora. So these patients has very different, very rigorous changes in the dynamics of their intestinal microbial flora. And in memorial cell cluttering, what we have for the patient is that about 7 days before transplantation, they start to have the conditionings and start to generate the neutropenia time. And meanwhile, these patients are required to have this antiprophylaxis. So According to the procedure of the LOHCT, we can see that the patient will have very big changes in the microbial flora composition and the dynamics at the early stage of the transplantation. Therefore, we study the fecal samples collected between day -10 to day 30 in these patient cohort. So I'm not going to talk about too much about the general view of that dynamics of intestinal microbiome and the clinical outcomes, because my colleague, Dr. Jerry Rowling, is also going to give a talk in this seminar series later this year. Please stay tuned for that. In today's talk, what I'm going to talk about is more like my experience in the microbiome studies and some, I think, very interesting and important data, but could be overlooked in the publications, especially the approaches, how to study the microbiome. When I start to establish the microbiome pipelines in the Tobias Holt's lab, I borrowed a lot of ideas from the bacterial world. Even till now, there are three major approaches to study the bacterial microbiota, which includes first the functional analysis, that is the multi-omics, and the DNA-based genomic analysis, including 16S and the metagenomics, and the classic microbiology culture method, that is to isolate the organism for the phenotypic and for the whole genome sequencing analysis. What can we transfer for these approaches? What can we transfer for the fungal field? We pretty much cannot do much functional analysis with the multi-omics because they're not very good pipelines, but we can do the genomic analysis and the cultural omics. So in the fungal field, in the microbiome studies, the ITS sequencing is still the most widely used method to characterize the composition of the fungal flora, and it can do it can provide high throughput results to give the researchers a general view of what is the composition of the fungi are. And recently, we start to see the metagenomics pipelines to assemble fungal genomes from the shotgun data. This still probably need more improvement, but it's coming. And also, we can do the cultures. So I'm going to show you some of the data that using different methods and what can we learn from the different approaches. So by ITS sequencing, what we learned is that in a case control study with candidemia patient is that in this patient, before the bloodstream infection being identified, about a week before the bloodstream infection being identified, the patient start to have the intestinal domination with Candida, with pathogenic Candida species. And therefore, by this sequencing, we suspect that the origins of the bloodstream infection is from the gut expansion of the Candida species. This is by sequencing. So what about by the organisms, by the microbes? I'm going to focus a lot on this patient 3. And some of you may notice that I kept on saying Candida parapsilosis complex species. Why I say that? This is because the clinical micro labs in Memorial Sloan Kettering Cancer Center told us this patient had 3 positive blood cultures on day 13, day 14, and day 16, and they are all parapsilosis. This has been identified by the Maditov method. And meanwhile, I also isolated Candida strains from the fecal samples from this patient. And I sent those fecal strains together with bloodstream strains for whole genome sequencing by expecting that they could share very similar genomes and to build up the linkage between the fecal strains and the blood strains. So we sent these strains for whole genome sequencing, and we collaborated with Dr. Jaren Butler and her student Ella for the whole genome sequencing analysis. But later, Ella gets back to me saying that 2 out of the 3 parapsilosis, 2 out of the 3 bloodstream isolates are actually orthobacillosis. They're not parapsilosis at all. And in the hospital, and differentiate the 2 sister species. And this makes me feel very shocking because you can see this patient fecal samples is kind of full of parapsilosis sequence. And where is orthobacillosis? It's a little bit here, very, very low amount, relative abundance. It turns out that in the 5 samples here, there are about 2.2% to 1.6% of the fecal sample sequence belongs to the Orthobacillus. And in order to build up the linkage between the gut organism and the bloodstream infection, I need to isolate the Orthobacillus sequence from the gut samples and compare the genome sequence with the bloodstream isolates. Therefore, I screened over 500 samples and do the genotyping and found about 1.4% of the strains I isolated are Orthobacillus. This ratio feels perfect in the range of the ITS sequencing pipeline. So by the culture method, I kind of validated the robustness of the sequencing pipeline. And with the whole genome sequencing of the gut Orthobacillus strains and the bloodstream Orthobacillus strain, they are very similar to each other, and we did build up the linkage. And we want to wrap up the Orthobacillus story, put it aside, and now focus on the parabacillosis compartment. So as I said, there are, I also isolated parabacillosis strains from the fecal samples in this patient and did whole genome sequencing and found that the fecal parabacillosis strains are actually fall into two different clusters. They're different two clusters. And by ITS sequencing, we cannot, at that time, the sequencing method, we cannot identify the difference between the two cluster strains. However, when we look at the ITS amplicon sequence, there are indeed one base pair difference. Why can't we detect the difference between them? This is because the method we use at that time is based on the OTU clustering method. The operational taxonomy unit cluster relies on to compare the sequence and cluster them with 97% similarities and calculated a similar sequence, calculated a representative sequence for the taxonomy detection. And by definition, if two sequences has less than 3% difference, this OTU clustering method cannot detect the difference between them. Therefore, we cannot detect the two parabacillosis strains difference, cluster strain difference. Luckily, in 2016, Ben Callahan from Susan Holmes' lab developed this data2 algorithm to process the raw sequence into the ASV, amplicon sequence variants. These are the actual sequences and can detect a single base pair difference. Once we switch the data to this data2 based pipeline, now we can detect the ITS sequence dynamics correlates with, which echoes the dynamics of the whole genome sequencing of the strains. That we can see the ASV1 sequence pattern that is carried by the cluster 1 strains shrinks over the time, while the ASV2 sequence expand, that the ASV2 sequence is carried by the cluster 2 sequences, cluster 2 strains. So now maybe we start to think about why there's this shift between a dominated ASV1 strain, the cluster 1 strain, to the cluster 2 strains. There is a panel that I kept on showing, but I haven't really explained it. That is the antifungal prophylaxis. We can see that during the sample collection, the patient was constantly on the mycofungin prophylaxis. So naturally, I start to ask, are there any drug sensitivity difference between the cluster 1 strains and the cluster 2 strains? I performed a e-test analysis assay, and I found there's barely any difference between the halozone size and there's no tolerance colonies such as the what we observed in the azole e-test strip analysis. But when we use a more rigorous test, that is to examine the survival rate at different drug concentrations, we can see that the cluster 2 strains can maintain a small subset of cells viable even at very high mycofungin concentrations. Therefore, the mycofungin prophylaxis may provide a selective pressure towards the cluster 2 strains and let the cluster 2 strains win against the cluster 1 strains. So with this data, I can show you that we can identify the strain level dynamics of microbiomes by the ITS sequencing, as long as there is a sequence difference in the ITS1 region, and of course also by the culturable fungal strain to compare the culturable fungal strains. And in fact, we collected over 2,000 fecal fungal strains from the LOHCT patient cohort, and these are very valuable resource to study the fungal pathogenesis and the microbial ecologies. And with this resource, we observed these strain level dynamics of microbiome can happen with many fungal species, including Parapsilosis, Labrata, Crucii, Saccharomyces cerevisiae, etc. And what's more interesting is that I found in the patient with Parapsilosis bloodstream infection, they are more likely to have co-colonization with 2 or more different Parapsilosis strains compared to patient with Parapsilosis intestinal domination, but without bloodstream infection. This is very interesting because these patients with or without bloodstream infection, they are all LOHCT patients. They live in the same unit of the hospital. So technically they have the same access to acquire the environmental Parapsilosis strains, but why some of them had monocolonization, but some of them had co-colonization with different strains. So this colonization status difference may reflect different metabolic status of the Parapsilosis strains and may further reflect the different status of the host immune status and the bacterial fungal interactions in the same niche. So I know these are very hand-waving messages, but I think this is very interesting and worth further investigations. Okay, so taken together, I'm going to come back to the 3 keywords in the microbiome study: the context, approach, and implication. Now we know that there's when there's a robust intestinal colonization with fungi, it's always been associated with these different disease conditions, such as LOHCT, IBD, alcoholic liver disease, etc. And what we really need is we need more human data to plot a more comprehensive view of the dynamics of microbiome and to learn what happened in different pathological, physiological conditions in humans, what the role of fungi is there, and then provide the potential future directions for the fungal biology and fungal pathogenesis study. And in terms of approaches, we can use kind of a robust ITS sequencing pipeline together, the whole genome sequencing pipeline, whole genome sequencing on fungal isolates. But we really really requires more better approaches for the microbiome analysis. For example, for some specific diagnosis purpose, for some specific pathogenic strains, we may need better amplicon resolution. So we may borrow the ideas of the loop-seq from the bacterial sequencing, which can sequence the full length of the 16S rDNA region and provide a higher resolution. And we also need to develop the functional analysis pipeline, such as better metagenomics pipeline or metatranscriptomics to learn the functions of these fungi in the gut flora. And we also know that intestinal fungal colonization may lead to the bloodstream infection, also may exacerbate the primary disease such as inflammatory bowel disease. And this brings up the questions for the requirement for the mechanism studies, and such as Kyla study recently focus on the fungal host interactions, and we also want to know what about the fungal bacterial interactions in the same niche. In the end, now we know that the microbiome could link to different disease conditions. Can we manipulate that to improve the clinical outcomes, which also requires further investigations? Okay, with this, I would like to thank my mentor, Dr. Tobias Ho, and my colleagues, Thierry, Mergen, and all the members in the whole lab. I really enjoyed my time in New York and in Memorial Sloan Kettering. And I also want to thank my collaborators all across the world. This is a very interdisciplinary project, and without any one of you, I cannot achieve this. And in the end, as I said in the beginning, I just started my own lab, and I'm actively recruiting. If you have interest in study the fungal microbiome, please contact me. Thank you very much.
Thank you, Bing. That was wonderful. We'll give everyone a few minutes to get questions into the chat. There are a couple that there already, and I'll give the next question to Shelby and then hand it over to Liz. So Shelby, your first question comes from Matt Blango. And he says, awesome story. I am wondering if loss of ZNF is necessary or if transcriptional downregulation could be an alternative strategy that you might expect to observe in some of your other hypermutator strains.
Yeah, I think loss or transcriptional downregulation of any gene required for RNAi could mediate something similar. We did also do nanopore sequencing for 2 closely related non-hypermutator isolates to look at CNL1 in those strains. And we do see CNL1 subtelomeric arrays in those strains as well. They're just not as large in the non-hypermutators. And then we also have some chromosomes in those non-hypermutators where they don't have the arrays. So they definitely have the propensity to become hypermutators if they lose RNAi function in any way.
You're getting a thumbs up. I think you can see that.
Oh.
If you can't see
Oh,
that.
yes, I can.
Okay.
Thanks, Matt.
And then, so I'm very impressed by the amount of subtelomeric rearrangement you see. And so I was wondering whether you also saw changes in things like the level of the sirtuin proteins in the subtelomeric regions. And so do you get changes in the regulation of the transcriptional silencing?
So we haven't looked very closely at changes in overall gene content, and we also have not done RNA sequencing to look at changes in expression for any genes. We've only done the small RNA sequencing. So unfortunately, I don't have an answer for you on that question, at least not yet.
And then how often do you see, say, resistance to multiple drugs in a single colony? Because I know you looked at 2 different drugs and split the population. So you have one, it sounds like you have one mutation that is consistent, that is a single gene mutation that can confer resistance to FK506. But are there other possible mutations, I'm sure drug pumps would, that would confer resistance to multiple drugs? Or do you even have something like cross-sensitivity where if you're resistant to one drug, you're sensitive to another?
So I haven't like specifically, let's say, plated like resistant colonies from one drug to another drug medium. I have tried to take like a rapamycin FK506 resistant colony, I see Bing is back, and tried to plate that or like grow up a culture and see if I can get like a multi-drug resistant colony or isolate. And I haven't been successful with that yet, but in theory, there's no reason why there couldn't be multiple insertions of CNL1 throughout the genome that could mediate resistance to multiple drugs, whether it's multiple insertions in different genes, or if you have an insertion in something like a sterile biosynthesis pathway gene that could confer resistance to fluconazole and then maybe also decrease sensitivity to amphotericin B. So it's definitely possible. We just haven't seen it yet.
All right, we'll do at least one more question while Bing's getting set up. Sean Beckwith says, great story. I'm wondering how widespread this mechanism of drug resistance by transposon activation may be. Is HN99 unusual in having no full-length CNL1? And what is the copy number distribution of CNL1 in the collection you have sequencing data for?
So how common is it? It's not common from what we can see. However, I will say that we've looked at CNL1 abundance throughout all those strains from what we can tell based on the Illumina data that we have for all of them. And CNL1 does seem to be distributed throughout all of the different lineages of C. Neoformans, and it was also initially identified in the sister species Cryptococcus Neoformans. In the VN1 lineage, which is where H99 is located, fewer strains have CNL1, so I don't know that it's unusual. It seems to be, it kind of goes both ways. It's possible that maybe having a full-length copy of CNL1 might be more unusual, but it's definitely found in all the lineages throughout. And then I can also read the second question if you want me to go ahead.
Yeah, why don't you go to the second part of that question?
Conversely, how common are mutations in the RNAi genes, and so on and so forth. So we've been developing pipelines to take that publicly available Illumina sequencing data. Unfortunately, when they posted that data, it's just the raw sequencing data. So it's been kind of a journey to get assemblies for all of them and looking through all the genes. I will say we have identified a handful of other RNAi mutant candidates, and we're investigating those now. So there are others that have nonsense mutations in those or frameshift mutations. Those are what we've looked at. Just, it's the easiest thing to look at first. So there are others, and it'll be interesting to see if they're hypermutators. And we think that'll also kind of be dependent on their CNL1 burden.
So Shelby, your question is from Liz, and she was wondering about the mating competency between the BT strains and H99, given these large differences in transposon occupancy. Do you see impacts on viability?
So the two strains are actually MAT-A, so they are mating compatibility with H99 as well as 99% of the other known clinical and environmental isolates of Cryptococcus neoformans. So there's definitely a great, they're mating compatible with a ton of Cryptococcus strains. They have about a 20% viability rate from across. We don't think that's due to the transposon burden. We actually think that's due to the two chromosomal translocations between those strains. So each translocation is expected to drop at about 50%. So 20% is about what we would expect. So yeah, technically these two MAT-A strains could mate with a ton of other strains in the environment, and these elements could spread. However, it would just be dependent on whether or not they have RNAi, if they can be active. Thanks, Liz.
Great, I'll hand it over to Liz for a few questions for Bing.
Thanks for the answer to that question, Shelby, that was great. So Bing, Jessica and I have asked basically the same question, but I'll ask hers first and then I'll ask mine second. So she wants to know, are transplant patients infected by less common Candida species, such as, are they frequently infected by the same strain? Do you think they are acquired from the hospital, or is it coming from their own microbiome?
That's a great question. So first of all, the common colonization with parapsilosis in Memorial Sloan Kettering is very likely associated with the Mycology prophylaxis, because we know that parapsilosis species are more tolerant to mycofungi, echinokinase in general, compared to albicans glabratus. So this is a kind of a context-specific issue with parapsilosis. Regarding strains, yes, I think after I reviewed a lot of patient chart, I think there are definitely patients acquired the strains from the hospital. I cannot say all of them have the parapsilosis, acquired the parapsilosis in the hospital, but I think definitely there are connections, we found the strains between the patient. They are very similar to each other. There's potential transmissions between patients, yes.
I think that's really important. During the Candida auris outbreak, there was lots of effort to track exactly who was passing it between.
Yeah.
Obviously, that's not happening in the case of parapsilosis, although it's been much more present for much longer time, right? I was really surprised by the diversity of the strains that you're seeing, because it's not just one isolate that's being passed around the population, right? There's actually enough diversity that you're able to even detect it just by ITS. That says there's a lot of parapsilosis.
Yeah, it's kind of lucky that there's a sequence difference in ITS region, so we can detect that. If I use because I use ITS1 for the ITS sequencing, if I use ITS2 as a lot of labs, I cannot detect that, to be honest.
Yeah, no, and I think that's really, it's lucky, but it's also like, well, is it evidence of a lot of diversity in the hospital compared to, or is it coming through some environmental into the hospital? So for example, the way mucorales infections happen, where they're coming from the outside into the building. Do you have a sense for where that reservoir might be?
No idea.
No idea.
No idea. Yeah, I didn't sample any environmental. I didn't do any environmental sampling, so that's something I want to do, but because of COVID, because of different things,
This is the reality of 2021.
It's very difficult, but I think it's really valuable because to investigate the environment of the hospital, because as I think Julie Segre's lab published this bacterial transmissions in the NIH hospital, that they sequence the plumber, like the pipes of the hospital, and find the bacterial pathogen from the environment, and transmissions from the two different patients. So that's definitely a very valuable, could be very informative if we do that part of research, but need a lot of effort also. And
yeah, of course.
I wish I can have another year in the MSK.
I think this is all of us we could go back and do our postdoc for one more year just to answer some of these questions.
Yeah,
I got one more question lined up, but do you want to pass it back to you and you can ask more for Shelby, and then we can finish up with Bing?
Sounds good.
Okay. Do you have one more for Shelby? Sorry.
Oh, sorry.
I can read it if you want.
I misunderstood. So Saya Gusa asked, do the hypermutator strains show any fitness defect, such as a high CNL burden? I know you mentioned that you're planning to follow up on fitness, so you might not have done this yet.
Yeah, so we haven't done it yet. I will say the two hypermutators seem to grow a bit slower in YPD, and they also seem to have a hard time growing in YNB medium that's not supplemented with amino acids. So at least in laboratory environments, they might have a fitness defect. We don't know, I guess, how that would impact their fitness, say, in the environment. And they are obviously clinical isolates, so they are fit enough to infect humans, but I don't know relative to others how that fitness changes.
I guess we do have more amino acids lying around in our tissues than is perhaps comfortable.
Yeah, some growing them for chef gel analysis, we had some issues, and it was specifically with BT65 and BT81 and hypermutator progeny, but it wasn't an issue for non-hypermutator progeny or non-hypermutator closely related strains.
There's some really disturbing implications from that.
Oh, yeah, definitely. What's interesting is in the progeny, we didn't see that much CNL1 movement into other regions of the genome. There was a lot of movement in those subtelomeric arrays, and then obviously from our antifungal drug assays, they can move into drug target genes, but it doesn't seem like they're randomly moving everywhere all the time, I would say.
And then there's a question from Alexander Bruch, who says, great talk. Maybe I missed it in the talk, but is it known, or did you investigate at which level RNAi silences CNL1? Is it happening by transcriptional, heterochromatin formation, or translational silencing?
For some transposons in Cryptococcus, we know that that they are actively transcribed, at least during mating, and then that they're post-transcriptionally silenced by RNAi, where they're going to have their messenger RNAs cleaved by the RNAi machinery. And then for a lot of other centromeres in Cryptococcus, like the centromeric ones, and then also at some subtelomeric regions and other regions, we know that either 5-MC DNA methylation or other heterochromatic marks tend to localize with transposons. But I think at least for CNL1, I think it would be post-transcriptional, but both transcriptional and post-transcriptional are known in crypto.
Great, thank you very much. And I'll pass it back to Liz for questions for Bing.
Yeah. So I'm just going to keep asking you questions until there's more put in the chat, but this is fantastic. I'm really excited to pick your brain about this. So I wanted to get back to this peropsilosis, orthoxilosis thing that you observed, where it seems like one species is more prone to bloodstream infection than the other. Is there anything more that you can tell us about that dynamic?
You mean the orthopsilosis and the peropsilosis, or
yeah? So is it something about morphology? It seems like there's an event that's happening where it's escaping from the gut and getting into the bloodstream, right? And do you think that's
so?
One
thing is that what we sequence or what we analyze are the fecal samples. Right. These are the shedding materials from the gut moving, and what happened really at the mucosal barrier could be very different from the shedding materials, right? So I think I had some very preliminary data that I did a attachment analysis of the strains to the gut epithelial cell lines, And I found that orsopsilosis is more adhesive to the epithelial cells compared to the parapsilosis strains in the same patient. So I think that's probably more likely the reason why we see orsopsilosis in the gut, in the bloodstream, with such a low frequency in the gut abundance.
Yeah, no, really, really interesting. Do you have any comments on what changes in bacterial populations might be doing in relation to these two organisms?
You mean in terms of the orsopsilosis and parapsilosis?
Yeah.
Actually, I mean, this should be Thierry's talks topic, but I just give it a teaser answers here. Actually, when we found parapsilosis or orsopsilosis, when we found pathogenic fungal expansion in the patient's gut, it's very likely that we see very low bacterial abundance. So it's kind of like we need very low bacteria in the gut to have the fungal chime in and expand there. So it's difficult to say which exactly the bacterial players associated with different species, but I would say more like a general, general trait for the fungi to be able to colonize in the gut, that is to have a bacterial reduction. Yeah.
Okay. So just as more information. Yeah, absolutely. Yeah. I completely agree with that. Absolutely. Jess has one more final question, which is, she says, I'm very curious about Parapsilosis' tolerance for mycofungin. Is this a clinical tolerance or rather than true resistance? And are you demonstrating clinical importance of this concept of tolerance?
So yeah, I want to clarify that. The tolerance is just like a relative speaking. I mean, people still on clinical still use echinocandins to treat parapsilosis infections. It's just for mycofungin itself, the definition of breakthrough concentration is different. So for albicans, the breakthrough concentration is 0.25, but for parapsilosis is 2 microgram per milliliter. So it just have different breakpoint concentrations, but we still in clinic, people still use echinocandin to treat that. It's not how to say, we just say parapsilosis is non-treatable by the echinocandins. It's it's just relatively more resistant, but not a absolute resistance events.
Yeah, I just find this very exciting because there's been a lot of work on tolerance, such as Judy Berman's work, but the clinical relevance of tolerance has been under debate. So you might have some clinical relevance of tolerance here.
I'm working on that. Hold on.
I look forward to seeing it.
I want to have a more definitive answers for that in the next few months. I may have more definitive answers for that. Yeah.
Excellent.
All right, thanks everyone. We'll see you next month, and hopefully we'll see you at the Mycotalks on August 26th as well. And thank you to our 2 fantastic speakers.
Yeah, thanks so much, ladies. That was a really, really fascinating section.
Thank you for having us, Jessica and Liz.